Okay, hi folks. I know some of you have midterms coming up, and I don't want to keep you too long. So we're going to go through a couple of things really quickly today about schizophrenia. I'm actually setting up the paper that many of you have been hearing a lot about, and looking at the MHC complement C4 system and how that actually might play a role in schizophrenia. And I wanted to take you through schizophrenia first, and I wanted to give you a framework for why this genetic study is actually so powerful, and potentially why it's going to be a game changer. It's already been touted as one of the most important papers related to schizophrenia that's been published in the last 40 years. And that's actually a really big statement. And I'm going to take you through a little bit of the new research ideas. But I wanted to bring everyone up to speed. And again, this lecture isn't on your midterm, but I wanted to bring all of you up to speed on schizophrenia, some of the research that we know a little bit about, before getting into the genetics after the midterm. And Thursday, what we're going to do is have the entire tutorial devoted to getting you ready for the midterm on Thursday. So we'll go through some practice questions. We'll go through any of the questions that you have. So bring your questions to class, as well as going through specific slides, lecture by lecture, that you need to know for your upcoming midterm. Schizophrenia, as I'm going to mention, is actually one of those disorders that we kind of know a lot about, but in many ways we don't know a lot about it. And it's a very interesting disorder from different viewpoints, whether it's from a genetics viewpoint, or from a neurobiology viewpoint, or from a physiological viewpoint. This is such a complex disorder, but it's also such an important disorder that I want to take you through this. Now I'm going to just mention this as sort of as schizophrenia. I'm not going to talk about the schizoaffective types. Many individuals have different classifications. I'm just going to call this generally schizophrenia. I'm going to take you through a little bit about what we know about schizophrenia, and then why we think the way that we do about schizophrenia, and why that might be wrong. So when these original papers, and this is not the first paper that came out on the MHC class proteins, and it's not the first genetic study that implicates the complement system, but a lot of people were looking at that data going, that doesn't match anything that we know about the disease state. Why is that particular group of SNPs coming out as being positive? So I'm going to take you through some of the inappropriate parts of the framework, why that has actually thrown us off for a very long period of time, and then how we're going to look at the genetics of schizophrenia in this particular paper that I'm going to introduce to you. Okay, so schizophrenia, we know a lot of things about. We know that it is heritable. You know that if it's in your family, that if someone in your family has schizophrenia, that there is a likelihood, an increased likelihood, that you or someone else in your family will also have this psychiatric disorder. A lot of you will sort of characterize schizophrenia as delusional, or having a psychotic state. Definitely happens. However, for the most part, we're looking at impairments in cognition. All the things that you hear about in schizophrenic voices, for example, that happens, but the changes in perception are actually usually preceded by changes in cognition. You have problems with paying attention, you have apathy, you have a lot of cognitive deficits before you have things that are wrong in terms of the stereotypical things that we associate with schizophrenia. We don't really know the pathogenic mechanisms. When you take a look at someone's brain that has schizophrenia that's passed away, we know that there are structural differences in the brain. We know through imaging studies that there are pathological differences, but we don't know the mechanisms that cause schizophrenia. So this, again, is a very interesting disorder because of that. And we know, for example, and I'll show you some slides related to this, that there is loss of gray matter, that there are overall reductions in the synaptic structures. So we know it is a physiological disorder that affects the brain, but we don't really understand how it comes to pass, okay? So we also know that there are treatments available. And in fact, this is where the biggest issue with neuroscience comes in. We have drugs that actually make some of the symptoms in some forms of schizophrenia disappear. If you're given, for example, chlorpromazine, or you're given haloperidol, some of the symptoms of schizophrenia disappear. And as a result, that's actually set back our understanding of the disease and this disorder for a while. So if you have a cure, in other words, you think you understand the mechanism, and you're looking in that particular region. And I'll get back to this when we go over dopamine. However, even though we have drugs that may treat some of the symptoms of schizophrenia, we don't really understand how these drugs work. And I'll go through some of the data with you as I go through this. So we have some effective therapies, but they're not working 100%. It's a lot like looking at a mood disorder like depression, where just because some different drugs that are used to treat mood disorders work, we don't really know the mechanism for how things like depression occur, and it's a lot like what we see with schizophrenia. Because the drugs seem to relieve some of the symptoms, we think we understand the mechanism, but in fact we really don't. So overall, one of the more important things is, in genetics, we want to understand sort of the foundational way in which this inherited disease actually works, and we want to understand how this works, particularly in the biological processes that I'm going to describe for you. We need to understand the mechanism, in other words. So even though we have drugs that relieve some of the symptoms, even though we think we know that there are structural changes, we really don't know a lot about the disorder, and we need genetics to actually help us to understand it. And I'm going to try to give you that framework in terms of this new paper that has come out. Schizophrenia, we've talked about things like temporal lobe epilepsy, and we've talked about things like muscular dystrophy and other things. But in terms of the prevalence, how often you see schizophrenia, it's actually much, much more prevalent. It's 1 out of every 100. So think a little bit about that. So 1 out of 100 individuals at some point in time will be affected by schizophrenia. This makes this one of the biggest sort of disorders that we have out. And when you see this, and usually you see this if you're in a public space and you happen to be riding public transit, or you're downtown, you'll see someone who has some of the different sorts of positive symptoms of schizophrenia. They might be shouting, delusional, psychotic, whatever you want to call it. Usually those are the scarier symptoms. But in fact, even if you don't see those, there are probably individuals that are going through schizophrenia as this is a very prevalent disorder. 1 in 100. It means that at some point in this class, you're going to encounter someone who's got, who's had schizophrenia or these schizoid spectrum disorders, okay? So that's one of the reasons why we're really interested in this. The other reason is the biology is so fascinating too. Males and females are almost equally affected, but the onset in males occurs earlier than it does in females. Males get affected first, and then females usually get affected a little bit later. But both of them, males and females, tend to get affected. Do you know what this term refers to, a prodromal period? Yes. So you have mild symptoms or almost no symptoms, and that is the prodromal period. So milder symptoms, not the really prevalent type. So you might not be able to concentrate as well at school. You might not be as motivated as you were at school. And these might be sort of the prodromal periods before you get the full onset of the psychoses, the delusions, the hallucinations, and everything else that is very common in schizophrenia. So again, some of these cognitive changes that occur, so these are the ones that precede the positive symptoms that I'll get to in a moment. So some of the cognitive changes might just simply be having problems at school. You had a student who has been doing really well, and then suddenly at school they aren't doing as well, or they might be having difficulties while they're sitting in class, not being able to concentrate, not being able to really follow what the person at the front is saying, and they're having some issues in terms of their cognition. Eventually, as this progresses, they might start having that sort of prototypical suspiciousness of others or mistrust of other individuals. This is often the change in perception that I mentioned to you earlier is often one of the prototypical, one of the characteristic features of schizophrenia, and things may sound or appear different. Do you know what the first most commonly recorded or most commonly reported change in perception in individuals with schizophrenia is? Yes.
I think it's hearing sounds.
So hearing sounds for sure, definitely. Usually hearing sounds occurs, but what kind of sounds are reported first by most individuals that have schizophrenia? Yes?
Sorry.
Yeah?
I'm thinking alarms are things that you normally hear on a day-to-day basis.
Actually, it could be things that you hear on a normal day-to-day basis, yes.
I think it's voices in your head.
That comes, definitely. Voices in your head, hearing voices. You might remember a few years back about the incident on the Greyhound bus where there was a passenger seated at the back of the bus who beheaded the person that was sitting beside them on their way to Saskatoon. And he definitely heard voices. The voices told him to take the other person's head off. And that is schizophrenia. It's a very frightening disorder, right? So you could be thinking the person sitting right beside me on the bus might have schizophrenia. They might be hearing things and having this mistrust of other individuals that leads to them killing someone else. But what's the most common type of auditory change or perception that people have reported? Anyone recall? And it's kind of scary if you think about it. So it's knocking. So they hear knocking first before they hear sort of voices in their heads. They'll often hear knocking when they might be like in an open room like this. There's no reason to hear knocking behind you or beside you, and they'll hear sort of a knocking sound. So changes in the ways that things look or sound, which will lead to odd thinking or behavior, all of these sort of occurring together. Uh, Again, other things that will commonly occur, and again, you can understand possibly the reasons why. If you're mistrustful of other individuals or suspicious of other individuals, you might start to withdraw from family as well as from friends. You may not want to take care of yourself as well as you should, and you will also have that poor personal hygiene that is often characteristic of schizophrenia as well. So it's a very insidious disorder. 1 in 100 individuals are going to be affected and can become very, very serious. It's not common that individuals with schizophrenia become violent, but it is something that can potentially happen. And this change in perception as well as the change in behavior is often considered to be one of the characteristic cardinal signs of schizophrenia. And we know that we can classify these, and this is sort of the psychology, psychiatry version of what schizophrenia is. The typical symptoms are described as either being positive symptom or a negative symptom. So both of these things are happening. So you gain things like hallucinations. So you haven't had that before, and now suddenly you start to hallucinate. You may also start to have delusions where you're not able to think in a normal, rational way anymore, and you start to have delusional thoughts. You will have really aberrant thoughts and thought disorders, and those are things that haven't occurred before and now become positive symptoms. Some things are taken away, and these negative symptoms, where you're withdrawing or feeling guilty and withdrawing, alogia or not speaking as often as you were, as well as these cognitive deficits, really make this an interesting disorder. So if we had one of those symptoms, it'd be easy to describe maybe what system in the brain might be involved. But having both of these things, both negative and positive symptoms being associated with schizophrenia, make this kind of an interesting circuit to examine. So this gets into why I mentioned at the outset that this is an atypical and inappropriate description of this disorder. So one of the things that has been known for a while, perhaps inadequately and perhaps erroneously, probably not erroneously, but it's like seeing something out of place and then thinking that that's what should normally be there. So one of the things that happens in schizophrenia is that there's some sort of change in the dopaminergic system. Your dopaminergic system in schizophrenia becomes very overactive. It becomes hyperactive in many cases. And this is one of the reasons why we consider the dopaminergic hypothesis in schizophrenia, and it has been the dominant hypothesis for the past 4 decades. I'm going to give you some lines of proof that this is actually really important, and this is why everyone was looking for a problem with the dopaminergic system. So when you found a new gene and it was related to schizophrenia, it had to be related to dopamine because we knew that there were problems with the dopamine system, or it had to be a dopamine receptor that was being affected, or something that bound to a dopamine receptor. If it was something like the immune system, it made no sense, and people kind of discarded it sort of as an afterthought until fairly recently. So some of the other major lines of evidence is that if you look at someone who has schizophrenia, and you give someone who has schizophrenia amphetamines, in about a third of the individuals that have been diagnosed with schizophrenia, they have uber schizophrenia. They will hear voices, they will have delusions as soon as you start giving them amphetamines. In some individuals who are chronic users of amphetamines, these psychostimulants can also produce paranoid psychosis. So they start thinking and being mistrustful of other individuals, similar to what is observed in schizophrenia. Why is that important? Because we know that amphetamines actually affect the dopaminergic system, and we know that after you take amphetamines, the levels of dopamine at the synaptic terminals increases. So if we have a psychostimulant that causes a psychotic behavior which increases dopamine, doesn't it make sense that this should be a problem with dopamine? And since we also have a third of the patients with schizophrenia actually getting worse after you increase dopamine with amphetamines, doesn't it make sense that it has to all center around dopamine? And again, with all of these sort of observational studies, it became more and more entrenched in the neurobiologist's way of thinking that it had to be something related to genetics underlying dopamine signaling, or a dopamine receptor, or a variant of this. So again, I know some of you have had this multiple times. I'm not going to ask you to memorize the entire dopaminergic circuit, but actually understanding what dopamine does in the normal brain helps us to understand why some of those symptoms actually might occur. So dopamine itself is a neuromodulator. It's not a true neurotransmitter. And this neuromodulator actually, in this particular diagram, we're not looking at the motor pathways that run from the substantia nigra, but I'm trying to highlight here the motivational pathways as well as the addiction pathways. The hypothalamic pathway is also not something that we're going to spend any time on. But if you take a look within this area of the brain, which is known as the midbrain, we have a small group of neurons, which is represented by these little clusters that you see here, that produce dopamine. So we don't have a lot of neurons, in other words, that produce dopamine. So this is one cluster that produces dopamine, and I'll show you this on another slide as well. And this cluster down here is another cluster that produces dopamine, and the substantia nigra is involved in the motor pathway. We're not going to talk very much about this, but this one down here, the ventral tegmental area, you can see that it sends its projections to these areas of the brain. And what areas of the brain are these? Where are they sending their projections, releasing dopamine in these areas? Yes, Richard? So this area is the prefrontal cortex, and we've examined this area previously as well, but it sends out dopaminergic projections throughout most of the brain, in the front part of the brain. Keep that in mind when we talk about the models for schizophrenia a little bit later on. Because of the way in which these dopaminergic projections, starting off in this little area like the ventral tegmental area, then sends its projections all throughout the brain into areas like the septum, in the striatum, and throughout most of the frontal cortex. Because of that, this may help to explain some of the cognitive deficits. If you're thinking about where it's targeting, where dopamine is being released, these are the areas that have often been associated with cognition. The other thing I'll mention in passing is we also get psychosis if you are given L-DOPA. So most of you know what we use L-DOPA for, and what do we use L-DOPA as a treatment for? Yep. So this is in Parkinson's, where when we have a dramatic loss of dopamine in the substantia nigra, in order to increase the levels of dopamine, we often give L-DOPA orally, and this L-DOPA overproduces dopamine until you get the dose right. Those individuals that take a lot of L-DOPA, they also have a psychosis. They have delusions, they have problems with cognition. So again, pointing to maybe dopamine produces some of the positive symptoms that are associated with schizophrenia. So far, our model is perfectly intact, that we have a problem with dopamine in the areas that I'm highlighting here for you, and that we know some substances like L-DOPA and amphetamines, which increase dopamine at the level of the synapse, will do exactly the same thing. Do you have any questions about this before we go on? And I'm not expecting you to memorize all of the structures here. It's just to give you a little bit of a background in terms of the neuroanatomy associated with the dopaminergic system. Yeah?
Dopamine is not responsible for the negative symptoms.
As far as we know, in an inadvertent way. And I'll go through the current model, and then we'll have to see in the paper whether or not they address this. And they kind of do. Any other questions? Yes.
Would usage of Adderall also show symptoms of schizophrenia as well?
There have been no reports that there have been any kind of hallucinations or delusions associated with Adderall use. But then again, the use of Adderall in a sort of recreational way has only been really popular for the last 12 years to 15 years. So give it some time, maybe. Don't take Adderall. Just study. Go to sleep, study, don't take Adderall. So again, I wanted to just point this out, and I need to point this out for a couple of reasons. When we talk about drugs of abuse in the next slide, the drugs of abuse that we're looking at, for the most part, not all of them, but many of them, things like cocaine, for example, all work to increase the level of dopamine at the synapse. So this is representing the synapse, and dopamine as well as amphetamine through different mechanisms, one through the dopamine transporter and directly blocking the dopamine transporter, making dopamine more available out in the synaptic cleft once it's released, or amphetamine actually working through a much more complicated system, but basically working the same way to increase the levels of dopamine out here in the synapse through a vesicular transport mechanism, and causing a competition for reuptake of either amphetamine into this vesicle or dopamine. And so dopamine takes this alternate path out. So either way, we're increasing dopamine at the level of the synapse. Why is that all important? Because when we increase dopamine at the synapse, some of that dopamine will interact with postsynaptic dopamine receptors. And we're going to talk about this in that paper. And some of these postsynaptic dopamine receptors, the ones that are found in this compartment, the postsynaptic compartment, depending on what they couple to, will either turn on the cell or turn off the cell. Okay, so if, for example, this neuron, this postsynaptic neuron, has the D1 family of dopamine receptors, and that includes D1 or D5, and they're considered the D1 type family. When dopamine binds to either D1 or D5, they will turn on the G protein coupling system that leads to an increase in cyclic AMP. And when it does that, it turns on the cell. That cell becomes activated, other ion channels become activated, this cell becomes activated, and it depolarizes. However, in some cells, when dopamine is released, it can also act on the D2 or the D3 receptor. Now, keep this in mind when I talk about dopamine a little bit later on. So D2 is normally the opposite. The D2, D3 receptor is usually considered to turn off the cell. When dopamine binds to it, if it's on that neuron gets turned off by reducing the level of cyclic AMP. One other thing to mention in passing, some of you will have heard of autoreceptors before, right? Have you heard of autoreceptors? So here is one instance where we have a presynaptic autoreceptor. So dopamine gets released here. Normally we think that dopamine gets sent out into this synaptic region, but in this case, this D2 autoreceptor will also detect dopamine being released, and it will turn off the synthetic pathway for dopamine. So if you get lots and lots of dopamine, this is one way to turn off dopamine production right through the receptor itself, the D2 autoreceptor. And again, there's a lot of controversy about this. Some papers, for example, say that if you get one shot of dopamine, that you can actually produce these psychosis, the delusions, the hallucinations. Most papers, though, if you read through them, actually suggest that it's chronic users that then get a subsequent dose that actually have hallucinations. I think the truth is somewhere in between, that there are some individuals that are likely going to respond to amphetamine very, very positively and have a psychotic event. But for the most part, it's not something that the first-time users really experience. So the truth is probably somewhere in between, and therefore the logic that amphetamines increase dopamine, dopamine causes psychosis, is actually somewhat of an erroneous observation. Again, I don't need you to memorize this particular diagram. All of these are different drugs of abuse. The VTA, or the ventral tegmental area, is one of those dopaminergic producing groups of neurons, which then send projections out to like the nucleus accumbens, as well as throughout the prefrontal cortex. If you take a look, though, do you see anything in common with all of the drugs of abuse? Any commonality with amphetamines, with things like opiates, barbiturates? Yes.
They all target the same place.
So eventually they all either enhance dopamine release, or make sure that we have more dopamine at the synaptic terminal, and it's all related to dopamine in one way or the other, absolutely. Anything else that you know about these different compounds? So things like amphetamines, opiates, cocaine, etc., THC, nicotine, all of these are plant alkaloids. So for whatever reason, our bodies are adapted to respond to plant alkaloids. These are naturally occurring substances. Plants produce these alkaloids. We use them because they increase dopamine in our brains. For whatever reasons, we have this mechanism in place within our own bodies. Okay, so again, if we go through some of the data, whether it's related to amphetamines or whether it's related to L-DOPA, elevating levels of dopamine in a normal, healthy brain may be equated with a psychosis, a hallucination, or a delusion. However, this is not exclusive only to schizophrenia. It's not the only disorder where we see this. We know that it occurs in Parkinsonian patients after L-DOPA, etc. But we don't actually know what the mechanism is. We can only describe that, yes, we see an elevation in dopamine. We have no idea why this occurs. We have no idea why it has an onset of 18 years of age in males and 25 years of age in females. And you have to wonder, how did we get to this particular model? So why this whole model that surrounds dopamine? I told you that this is the model that's dominated neurobiology for the last 4 decades. How did we get to that particular model? Any ideas before we get to the next slide? Are any of you really good? Yes, you have a theory? Yes, go. Yeah.
They found that what the antipsychotic drugs they had bound to the dopamine receptors.
Okay, that's actually correct. So one of the things, if you take a look here, these are different antipsychotic drugs that you take, and this tells you how well they bind to and block the D2 receptor. And all of these antipsychotic drugs, and the first one being found in 1952, haloperidol. Haloperidol, when you gave it to someone who had schizophrenia and they had delusions and they were hallucinating and they had all of the positive symptoms, if you gave them haloperidol and it was of a sufficient dose, all of those positive symptoms would disappear. And so they thought, wow, you know what? If this is working on the dopamine receptor system, therefore this disorder must involve dopamine. And all the drugs since then, things like chlorpromazine, even older class drugs like thorazine, all have been shown in one way or the other to affect the dopaminergic system. And so our original model came from studies that were centered around neuropharmacology. And again, you don't have to memorize these maps, but what it's showing you is all of these antipsychotic drugs that you see lining up here. Some are more effective at low doses, etc. Some bind better to the D2 receptor, etc. But all of them are really working at blocking the dopamine D2 receptor. And when you give them these drugs that are antipsychotic, as schizophrenics, all of those symptoms disappear. Okay, so this tells us that maybe changing the neurochemicals in our brain actually leads to a behavioral effect. And maybe everything in terms of behavior is related to neurochemicals and changing neurochemicals. It's not a very appetizing sort of theory, to be quite honest, and we know that that's wrong now, that it's not this simplistic model, but it was the one that came out because of the pharmacology that came out in the 1950s. Do you have any questions about this before we go on? Have you heard of any of these different drugs like haloperidol or chlorpromazine or different antipsychotics? Yes.
Isn't haloperidol also used for like a muscle laxant and sedative?
It can be in some instances, but it's not probably the haloperidol that you're thinking of. It is used for other purposes, but in this particular sense, it is a true antipsychotic. Okay. If we, for example, use what is known as a PET scan. Sorry, I'm just trying to get the laser pointer back up. I seem to keep losing it. Apologies on that. So if we were to use a PET scan where we have a radioactive substance, in this case, this radioactive substance is 11 carbon raclopride, and this is a radioactive substance that gets injected intravenously into your patient, and this shows that in an individual who has schizophrenia, certain areas of the brain really light up a lot, which means that these are areas where dopamine is being released a lot. Okay, and so when we take a look at the raclopride binding, which also binds to where dopamine is being released, that this is a really hot area in this particular individual with schizophrenia. If we give them haloperidol at 2 mg per day, and that causes about 74% occupancy of the D2 receptors, and then you go back in and see how much of this dopamine is really being affected in this individual. Because the D2 receptors are now being bound, dopamine can't bind and neither can raclopride, and we see that the overall levels of dopamine binding in the brain goes down, which suggests again, and this individual here on the right is also feeling better. They don't have the psychotic events, they don't have the delusions and hallucinations, they feel better after haloperidol, suggesting again it's all related to dopamine. There's no mechanism, it's just dopamine goes up, you get psychosis, we bring dopamine down, psychosis disappears, therefore dopamine equals schizophrenia. Okay? No mechanism yet. Any questions on this before we go on? And we call this positron emission tomography because when the radioactive tracer that we put in binds to its receptor, it gives off a little positron, which we can detect, and that's why it's called this positron emission tomography. The hotter, brighter color areas are the ones where we get lots and lots of binding of the particular tracer. For those of you who study pharmacology, just sort of out of interest, why does this particular slide actually mention receptor occupancy? Any ideas, or those of you who might be taking pharmacology class? I know some of you might be. No? So one of the more interesting things and why they still use these types of imaging studies, if you actually can make an estimate of the receptor occupancy with a particular drug, you can actually see if that individual will be responsive to treatment. So if you put in a drug like haloperidol and it blocks or shows a certain amount of blockage of the receptors, then you know that that person is going to respond to that drug. If they don't have that receptor binding, and blockade, they won't. And so this is actually a therapeutic tool that is used in pharmacology to estimate, is this drug actually going to work on this patient with schizophrenia? If we don't get above a certain receptor occupancy, this drug's not going to work. We don't have to wait. You don't have to mess around. You go to the next drug, like chlorpromazine. Does that work any better? If it does, great. We'll put that patient on that particular drug. So it seems like blocking those D2 receptors, whether it's postsynaptic, and there's a lot of controversy, is it the postsynaptic D2 receptor or the presynaptic autoreceptor that is being bound, somehow ameliorates or removes the positive symptoms associated with schizophrenia. We know again that if you are exposed to amphetamine, whether you're a chronic user or maybe you're one of those unlucky individuals that responds to one single shot of amphetamine and you also induce a psychotic state, it's also probably a dopaminergic effect as well. And we know that there are lots of pharmacological effects that dopamine might be involved in schizophrenia. Okay, but all of that actually is really misleading because it probably isn't related primarily to a dopamine dysregulation. That's really too simplistic. Is there any questions about this before we get into why it might be too simplistic? Yes.
I want to add that I think most of the dopamine receptor blockers actually make the negative symptoms worse.
They can in some individuals. That one of the reasons why we no longer call it schizophrenia and we call it a schizoaffective disorder. It's like any other kind of disorder that we look at. It runs a spectrum now, and there are some individuals that do respond and have more negative symptoms. You're right. Okay, so let me take you through like why everyone's so fascinated with dopamine, right? You keep hearing about dopamine, you keep hearing about serotonin, and this is one of the reasons why we think that dopamine actually might produce some of those symptoms that we took a look at. So this gets into when does dopamine normally work in your brain? What is dopamine there for? It's not there to respond to plant alkaloids, drugs, and to get you addicted to certain types of drugs. It's really there to signal. It's to tell your brain to wake up, pay attention. This is what you should be paying attention to. It's there to signal a novel stimulus. It's there to signal that there is a new or novel environment that you should be paying more attention to. It could be saying that, this is a good reward, pay attention to it. And so all of this is causing a dopaminergic neuron to fire more, or it could be an aversive stimulus, something that you don't want to be encountering. Dopamine will also fire there as well. This didn't quite work out the way that I wanted it to. Have any of you met Wolfram Schultz? He was actually here in Toronto about a month ago. He's still very active in research, and everything that we know about dopamine today came about because of his work in the 1970s. And so Wolfram Schultz is the first person to actually show that dopamine does all those things that I just mentioned to you. So at the time, he was working in Parkinson's disease, which is also related to dopamine. And one of the things that he was interested in researching in monkeys was to see, what do these dopamine neurons normally do? He thought he was in the substantia nigra, but it turns out that his recording electrode was just a little bit off. And so he wasn't quite at the level of the substantia nigra, but he was in sort of the dopaminergic midline neurons. So one of the things that he knew, and it was already known in the 1970s, is that when you give an animal a food reward, the dopaminergic neuron fires. It sends a little action potential. Okay, so you show the animal a piece of fruit, you have your electrode, you're measuring from a dopaminergic neuron, that's a reward, the dopaminergic neuron fires an action potential. Is that okay for everyone? So it is signaling for a reward, usually in response to food. That dopamine neuron fires in response to a food reward. But one of the interesting things that he saw, almost by accident, was that it's not just when the monkey sees the food or gets the food, it's actually a predictor of when this reward will occur. So I wanted to take you through this. Why is dopamine such a powerful signal in your brain? Why is it actually causing you to misinterpret things when you have too much dopamine in your brain? This is part of the reason why. So at the very top, what you have to imagine is like each of the points that you see up here in these darker bars, if you stack all of the different little dots that you see here, this is what you end up with at the top. So stack all of these little dots and you get all of these lines up at the top, okay? And what each of these little dots represents is the firing of a single neuron. So we get a little blip of firing here, some more here. We don't get many in this sort of empty region. And one of the things that he was looking at was looking in the dopaminergic neuron of a primate brain and seeing how does this dopaminergic neuron that normally responds to food, how does it actually fire? It sounds like a really simple experiment, but it's actually a profound experiment in many ways. So what you're seeing at the very top is without doing any kind of conditioning, all he is doing is recording from this neuron. So he's recording, he gets action potentials, action potentials, action potentials. He then gives a reward shown here on this line. And when he gives a reward, that dopamine neuron starts going crazy. It starts firing a lot more than it has before, which is represented by this big increase in firing. And we also see all these little points down here on the bottom going up as well, showing that this dopamine neuron that he's been recording from up until now, as soon as he gives a food reward, goes crazy. Do you all see that? Okay, so this is where dopamine is signaling for a reward, and it's very clear that when the reward is given, this surge of dopamine goes up. So you can also do this experiment where when you do things like a Pavlovian experiment and you give a conditioning stimulus, so not the food itself, but you ring a little bell and then you give the food. You guys know that experiment, right? So this is, he wanted to know, is it really the food? So the animal gets a food reward, the neuron starts firing. Is it really the food reward itself that causes dopamine, or is it the anticipation or the expectation of the food that causes this dopamine neuron to fire? So after the animal has been trained that every time you hear a bell, a few seconds later, you're going to get the food reward, he wanted to know, what happens to this dopaminergic neuron? So when you do this conditioning stimulus, there's no food actually given at this time. A little bell goes off, there's no food given. This dopaminergic neuron now starts firing like crazy in expectation that a reward is going to be given, and now it spikes. If we pile all of those little dots up, it spikes like crazy. No food has been given, no actual reward has been given, it's just the prediction of a reward. Do I have most of you so far, or I've lost some of you? So if you keep recording from this neuron, the same neuron all the way through several seconds later, and then you actually give that reward, do you see dopamine going up? Yes? No? So dopamine stays the same. It was really the expectation of the reward that caused this dopamine to fire. So if he stopped there, that would have been a brilliant experiment by itself. But what he did was even more brilliant. So he decided, what happens if no reward is actually given? So what happens to this dopamine signal if you give a conditioning stimulus, you ring a little bell, the animal is expecting to get a reward, and the dopaminergic neuron fires, and you can see it firing, but no reward happens. What happens, or what do you see happening to the dopaminergic neuron when it should be receiving the reward right here? Sorry? It stops firing. And so dopamine is signaling on multiple levels. It's signaling that you should be expecting a reward if you don't get a reward. So in other words, it is giving you salience, what is really important around you all the time. So imagine that this is the system that your brain is using to ascribe importance to things. And somehow you have an excess amount of dopamine and you're ascribing importance to this table. Is this table really that important to me? Probably not. But what if now, where I have lots and lots of dopamine that normally ascribes importance to things, that a reward is associated with this table, then now this table becomes really important to me because I see it with lots and lots of dopamine. And maybe this is the way in which the schizophrenic brain is at work. That maybe if there's too much dopamine, that this idea of this motivational salience, where a neutral event becomes really important to you, So maybe this classroom means nothing to you, but maybe coming to this classroom so that you learn something is important. But if you just ascribe importance to this classroom, that's that dopamine kind of taking over and prescribing a certain salience or importance to something that maybe shouldn't be all that important. And because dopamine is also there to help in the learning process, if you have too much of it, maybe you are misassociating things that normally you shouldn't be associating. And that was sort of the great idea that came about, and that's one of the reasons why with the pharmacology, with Wolfram Schultz and his idea that dopamine helps to signal important events, and you have too much dopamine, everything becomes important. That TV that you have turned off that seems to be talking to you, maybe one of the reasons why is you have too much dopamine. That dopamine is causing an inappropriate learning association as well as an inappropriate motivational salience. And so this became this amazing theory that had no scientific basis other than the fact that we have drugs that block, we have drugs that make you look like you have psychosis that affect dopamine, and dopamine has all of these features associated with it. Do you have any questions about this before we go on? I can tell you that this is a very scary disorder. I was sharing with Arjun just before class. I told you that this affects 1 in 100 individuals. So a few years back, about 6 or 7 years back, I ran into one of my old high school friends, and you know, we just got to talking like you normally do. I don't think you're at the age where you kind of run into your high school friends and just talk like, hey, what's so-and-so doing and how many kids they have. Not quite there yet, right? But we got together and we were talking, and I happened to ask him, whatever happened to my friend Ross? Like, he was like my best friend in high school. I kind of lost touch with him. Oh, he's in the hospital. I said, what are you talking about? So he had actually developed a really severe form of schizophrenia, where he believed that his TV, when it was turned off, was talking to him and it was telling him to do all sorts of different things. He had to get hospitalized because this became that serious. Now, I was thinking back a lot about that because, you know, you normally, you know, I still think of Ross in the same way, like my best friend. We had a lot of great times together, still the same person. But, you know, that schizophrenia, I would never have thought of all the people that I had as friends in high school, he would be the last person that I thought would ever develop schizophrenia. And he's still struggling with that to this day. And he will be struggling with that for the rest of his life. And it's one of those really, really scary things because, you know, you try to act really normal around someone that you know has been diagnosed with schizophrenia or any other type of mental health condition, but it's really hard to be honest. And you know, I think when he has a day where he's not feeling so well, because he lives fairly close to me, so I get to run into him every couple of months. You know, when he's not feeling so well and he's sort of a little run down as well, you kind of wonder if he's going into a schizophrenic state. And it is something that scares a lot of individuals. And he's had a lot of issues related to this. And I want us to keep all of this sort of in context, that this is a disorder that we want to understand for genetics, but it does affect people. It will affect you if those statistics are correct. And those statistics have stayed steady for 40 years. So there's no doubt that it will affect 1 in 100 individuals. And we need to understand the mechanisms. Okay, so I think now is a good time to take a break, and then we'll get into this model of what we think schizophrenia might entail in terms of a mechanism. Okay, if you don't mind, I'm just going to get going, and we'll see how far we get today. All of Thursday is just going to be a review to get you ready for the midterm. Some of you have been asking about the assignment. Some of you have been bringing up papers. Great choices in papers, by the way. But my first priority is just to get you through the midterm, the first sort of group assignment after the midterm. But I will be posting, as I mentioned, some exemplars for you to follow for your group grant, as well as the presentation itself. And again, I want you all to do fairly well. The midterm itself, I'll talk more on Thursday, but when I had my two graduate students that have been helping me out with this go through it, they did pretty well, and they don't actually take any notes or anything else, so I'm assuming that you'll all do pretty well on the midterm. on the following Tuesday. So we'll see how the review session goes. Bring your questions to class on Thursday if you can. If you're not going to be able to make it down, then you can maybe communicate with one of your colleagues or through the Facebook study group, and then someone can bring the question up on your behalf. So this is the situation that we're looking at. We have all of these different symptoms, including the positive symptoms associated with schizophrenia. We know that changes in dopamine experimentally in humans, as well as through different disease states, have also been implicated in psychotic-like events. And so this abnormal release of dopamine has been thought, because I just went through what dopamine normally does in your brain. This abnormally high release of dopamine in an unregulated brain, either through amphetamines or through schizophrenia for whatever reasons, the mechanism not clear yet, may change perception over time, because this leads to dopamine giving a sort of unique association or a novel association with whatever you were looking or paying attention to at that particular moment. And then if you keep having more of this dopamine release, it accumulates all of these abnormal events, and this may lead to, and this is a construct where this may lead to a cognitive scheme or a delusion to explain why that table seems to be so important in your life, because you kept looking at that table, you got lots of dopamine released from that table. That table must be really important for a reason. It must be important to you for a reason, and you're developing this cognitive scheme because every time you've looked at it, this table looks more and more important to you. Therefore, this table must have been dropped down here by aliens from another planet, right? And that's sort of a cognitive scheme or a delusion to try to explain why this table that you've been looking at with all that dopamine going off in your brain, why it's so important to you. Again, for most rational individuals, that makes no sense whatsoever. But for an individual that has schizophrenia, that makes perfect sense. That this dopamine, this inappropriate novel signal, this change in perception, why is that table suddenly so important in my life? All this could be due to this accumulation of dopamine. Those are the positive features that we associate with dopamine. I also told you that there are some negative features that are associated. So positive symptoms as well as negative symptoms. If you were to take a look at an individual and you were to look at the activity in the brain, and this region here on the left on all of these different figures represents the frontal lobe or the frontal cortex, and here's the occipital lobe in blue at the back. This is the overall level of activity. The brighter the color, the more activity there is. In a normal individual, looking at the left or right hemispheres at rest, the frontal cortex tends to be pretty active. In a schizophrenic individual, again, the activity tends to be lower at rest. In other words, the frontal lobe in a schizophrenic individual is low. It's not as active as we would expect in a normal individual. Do you know what this test is in psychology? Any of you who are taking psychology? Yes. Okay, this is the Wisconsin card sorting task. And during a card sorting task, like this Wisconsin card sorting task, in normal individuals, you see that there is an increase in activity in the frontal lobe. In a schizophrenic individual who is undergoing this card sorting task, it is not. And think about the task for a moment, right? So you're given this at the top, and then you're given this card here at the bottom. Right? And you're asked, put the card that you got down here at the bottom, put this card on top of any of the cards at the top, okay? And you do that, and you're not given any other instructions. So what if I put the card here? Would that make sense? Could, because it's red matching red, right? How about if I put it up here, 2 and 2, like 2 objects and 2 objects? How about if I put it up here? Positive signs, positive. So your brain is thinking, your frontal lobe is engaged. You don't know what they're expecting until they tell you, like they'll say, nope, that's wrong. So if I put this card up here, it's wrong, sorry. And then if you put it there, yes, that's right. So you have to match objects, right? And you'll figure that out fairly quickly. But in a schizophrenic individual, they can't do that. They have a really hard time in this card sorting task, this Wisconsin card sorting task. And we know that the frontal lobe is less active, and we call this hypofrontality. So yes, their brains overall have lots and lots of dopamine, but the frontal lobe is actually disengaged. It is actually turning off. And it's really hard to ascribe some of these negative symptoms with what we've just discussed. If really the dopaminergic system is going up into this frontal area, we would expect it to be much more active than it is. We know in structural studies that the gray matter volume also tends to be lower in these individuals. Yeah, Kay? They wouldn't be able to figure out the task. They would have a really hard time trying to figure out, like, I don't get it. What are you trying to get me to do? So if you follow the pattern really easily, as a normal individual, they would be able to pick up the pattern. So you then give them another card and ask them to put it on. They'll never be able to pick up what you want them to do, which is either match the number of objects or match the style of objects or match the color. They won't be able to pick that up easily. Okay. Any other questions? So we know that in these individuals, they have overactive dopamine, underactive frontal cortex, not able to cognitively engage. And I told you one of the signs that happens first is cognitive disengagement, where they're not able to have good cognitive thinking skills. The other thing that happens is in these individuals, if we were to take a look, that overall their brain, the gray matter tends to be dramatically reduced in these individuals as well. And it's not because of the drugs that they're given. So they are treatment naive individuals. They get diagnosed with schizophrenia. You put them in an MRI before they get their first dose of haloperidol, chlorpromazine, or whatever you're giving them as an antipsychotic. And these naive, treatment-naive first episode patients also have shrinkage in their gray matter. There are also differences in cell density, much smaller. The dendrites and spines that I'll show you in the next slide are also smaller at postmortem. So there is a structural change in different regions. And again, it's not just in the frontal lobe. So this is the dorsolateral prefrontal cortex. We also find it in the supratemporal gyrus as well. We find it in a number of regions in the brain. Throughout most of the gray matter, to be quite honest, if you're to take a look, the average reduction is across most of the gray matter, across most of the cortex. So again, it's a very interesting disorder. Where you see this much loss of gray matter, you'd expect to see all sorts of other changes, but we really see either positive or negative symptoms. And again, this represents at the top a normal individual's dendrites as well as spines. And each one of these little mushroom shapes that you see coming off of this dendritic structure that you see here is what we would normally expect to see in layer 3 of the cortex. And if you take a look at two different patients, so this is subject 410 and this is subject 466, you can see that in the same area of the brain, through Golgi staining, that we don't see that rich number of spines that we see up here at the top. So we know that there are severe structural changes that are going on as well, but we really don't have a good mechanism for why all of these things have occurred. So because of the way in which this sort of looks like it's occurring, that hypofrontality, the lack of frontal engagement, so the lack of activity in the frontal lobes, one of the things that we think happens is there has to be some sort of abnormality that hits this first. And I'll give you the rationale for why the frontal cortex has to be hit first. And again, just going back, remember it's the cognitive symptoms that are normally the prodromal ones, the ones where you're not concentrating well, you're not doing well at school, you're not doing well at work. It's those negative prodromal symptoms that we think lead up to the more massive positive symptoms that we think this hypofrontality usually precedes some of the more explosive positive symptoms that we see later. In theory, this is the neurodevelopmental model that we kind of have right now, that the prefrontal cortex actually controls the entire dopaminergic circuit, and the lack of control that we see in that hyperfrontality, so the lack of ability in the Wisconsin card sorting task, etc., that we think that this is why we have a dysregulation in the dopaminergic circuits within adults. And I'll take you through the rationale for that. It's not 100% agreed on by everyone. And in the paper that I'm going to be presenting after the midterm, that paper itself doesn't fully agree with all of the results that I'm going to be showing you here as well. So the theory is that somehow there is an abnormal development, and it doesn't explain where that abnormal development comes from, but it tries to explain the positive and negative symptoms. The prefrontal cortex, for whatever reasons, doesn't mature properly, and because it doesn't mature properly, it does not have as much feedback onto the dopaminergic neurons, leading them to have more dopamine release than they normally should have. Okay, and that's sort of the bare bone basics of this model that tries to explain everything. So this is the neurodevelopmental model, or also known in some places as the glutamate hypothesis, because all of this is explained through glutamate. Do you have any questions on this before we go on? If you understand sort of what I've just mentioned to you, the rest of it becomes a little bit easier. Any questions at all? Yes?
So this is basically just identifying the problem originates in the prefrontal cortex rather than the central human.
Right, so this is trying to use the prefrontal cortex and deficits in the prefrontal cortex to explain all the lack of dopamine. You're correct. And so I'll take you through this really slowly, and then this sort of sets up a couple of things on the next slide, which hopefully when I run through it twice, you'll understand a little bit more. One of the things that we know happens is that there is a feedback circuit from the frontal cortex, and it is a feedback circuit that goes into the dopaminergic neurons. This feedback circuit is controlled by NMDA receptors. This becomes key as well. It's an observational study, but it's a very powerful observational study that links some of the models that we've been looking at. And this might involve the cortical inhibitory feedback in one of the areas that involves dopamine, like the VTA. Okay, so this is just an overview slide. The next slide is the more important one that actually outlines the circuit from the cortex all the way down into the dopaminergic neurons. Okay, so this is the VTA or substantia nigra, either one. These are the areas that contain dopamine. And in the substantia nigra or in the VTA, the dopaminergic neurons release a dopamine, which is what you're seeing here. But here's this inhibitory system. This is the inhibitory system that actually begins up at the level of the cortex. So within the cortex, the cortical neurons send projections down, which is why when you're looking, we're always looking at prefrontal cortical pyramidal neurons. So if something's wrong with them, then we know that there might be an issue, and that's one of the reasons why we look at this in histopathology. Within the cortex, these glutamatergic neurons usually send their projections down to these inhibitory neurons, these GABAergic neurons. Okay, is that part okay so far? Because I'm going to take you through the circuit bit by bit. So these are the ones that sit up here in the prefrontal cortex. This is under normal circumstances. They will release glutamate. Glutamate binds to these inhibitory neurons and activates these NMDA receptors. These NMDA receptors then activate the inhibitory neurons, which then release GABA. Is that okay so far? Glutamate coming in, signaling through the NMDA receptors. These inhibitory neurons are now engaged. They turn off these dopaminergic neurons that release dopamine. So they're there to act as a control system. If I have too much dopamine, the cortex signals back through glutamate, through the NMDA inhibitory system, turn off, using GABA, the dopaminergic neurons. And as a result, less and less dopamine gets released. That's under normal circumstances. Is that okay for everyone? And one of the things that we think happens is that in some schizophrenic individuals, for whatever reasons, their NMDA receptors that are found on these inhibitory control neurons don't function well, or they're not there in enough numbers. Whatever the case is, now when that signal comes down here, these NMDA receptors are not activated. We don't activate GABA anymore. There is no brake on dopamine, and dopamine just continues to get released. And we release it to excessive amounts. Now, it could be for a number of reasons. This particular neuron up here may not have enough glutamate. It may not develop well. There's no glutamate to activate the NMDA. It might be a problem with the NMDA receptors on these inhibitory neurons. Whatever it is, it's this branch, the glutamate branch, not turning on this GABAergic control switch that leads to all of the excess dopamine that we see in schizophrenia. Is that okay for everyone? Yes, no, maybe? Do any of you need me to go through this again?
Yeah.
Thanks. So you could imagine, for example, that for whatever reasons, your cortical neurons don't develop. There's not enough of them, or there's not enough glutamate, and so it doesn't turn off this. It doesn't turn on this pathway that involves activating an NMDA receptor. So glutamate is released here. It binds to the NMDA. It turns on this GABAergic neuron. GABA then is released, and GABA then activates or turns off by binding to the dopamine receptor, the dopaminergic neuron, and no more dopamine gets released. And that's normally the brake that's on it. For whatever reasons, either you don't have enough glutamate containing neurons and it's underdeveloped, or there's a problem with the NMDA receptors. Whatever the issue is, we just don't have enough of a brake on this dopaminergic system. And that's one model that seems to hold up. And we have some experimental models as well. If we inject in ketamine into certain areas of the brain, one of the things that we see happening is when we're injecting ketamine, which is an NMDA receptor antagonist, blocking NMDA receptors on this pathway here, that we do see a rise overall in the release of of the dopamine in an amphetamine-dependent manner. So yes, it seems to model this, that with NMDA receptor blockade, if we don't get feedback, the dopamine levels do go up. So this glutamate pathway seems to be important in an animal model. If you use ketamine to block the NMDA receptor, it also in humans has been shown to induce psychosis. Nothing to do with dopamine directly, but here's another model of psychosis where blocking NMDA receptors also causes some of the positive symptoms associated with the schizophrenic model. So it appears to model both the negative symptoms and hypofrontality, as well as it tries to explain the overabundance of dopamine, but there is no full mechanism. Why don't we have that glutamate system in place? That's not clear. Why is there? Where does it develop? Does it develop? When does it develop? Also not clear in this model as well. And so this is where we're sort of at. We think we understand parts of it now. It's not just dopamine. It involves the inhibitory neurons that we just looked at. And this gets into the particular paper. Over the past probably 7 years, a number of different research groups have been looking at about 100 different loci in the human genome that contain single nucleotide polymorphisms. So one single nucleotide has been switched, and these haplotypes have been associated with schizophrenia. So we've known that there's a risk or concordance of these different haplotypes with schizophrenia for a while, for about 7 years or so. However, we haven't really been able to map out what all of this means. The functionality of this hasn't really been well described. Some of it is simply because these haplotypes that are mapped out run tremendous distances, like megabytes, megabases of distances to be able to map this out. One of these, again, you have to keep this in mind. In the mid 2000s, come up with some ideas that, you know what the problem in schizophrenia is? Has nothing to do with dopamine, has nothing to do with even glutamate, has nothing to do with dopamine receptors, has everything to do with your immune system and the complement system, the MHC that's found on chromosome 6. People were looking at that going, what are you talking about? How is that possible that we get these haplotypes occurring in schizophrenia? And it was a big mystery. People couldn't figure out like why this was the case. This article came about, and this is actually now considered already, it's only about 2 weeks old, it's considered a classic paper. It's considered a classic paper for a variety of reasons. One, it explains why having a complement system being activated might cause a change in development of your circuits. So it's a circuit model. It's not a neurotransmitter model. And so we know that this complement component in neurons, when it binds to complement component 3, C3, that it actually causes a reshaping of your synapses, much like I showed you earlier on. And I'll go through that a little bit later on. But this is actually a tremendous tour de force in the full sense of neurogenomics. It runs what is now considered traditional neurogenomics. And this gets into how this study was done. So this uses a genome-wide association type of study. And again, in 2007, these genome-wide association studies were considered the scientific technical breakthrough of the year. Everyone was publishing on these, where they were looking at haplotypes associated with these single nucleotide polymorphisms. It became really easy to do these types of experiments. As a result, everyone was doing these. And again, the overall methodology associated with these genome-wide association studies is you will isolate DNA from different individuals. You can then do either whole exome sequencing, which we'll get to a little bit later, or whole genome sequencing. Either way, a whole exome, of course, is much faster, but we can look at individual variation on this single nucleotide. We can see that some individuals have one version of the single nucleotide polymorphism versus the other. And we can start to see, how do these associate? When we look at the overall population, if we look at a population of schizophrenic individuals, do a cluster of these single nucleotide polymorphisms follow them versus the normal, the so-called normal population? And can we start mapping out where these single nucleotide polymorphisms are? And that's really where we've been stuck for the last 7 years or so. We've had a lot of information about this, but it's only that last paper that has really tried to address specific single nucleotide polymorphisms. And again, part of the reason why these studies are now so popular and so much more powerful than they were even 7 years ago is that the density of these single nucleotide polymorphisms chips are much more dense. We can put more of these different single nucleotide polymorphisms on one chip that allows us to do this much faster. So this doesn't sound, and I don't need you to memorize this slide. It's more for your own information. We have many, many common single nucleotide polymorphisms. We have to collect large numbers of cases, individuals with schizophrenia, in other words, versus like normal controls. And you have to think about what a normal control actually looks like. We then have to genotype all these individuals. So we have like 2,000 samples, let's say. That's actually not powerful enough. And modern statistics tells us, or population statistics tells us, that 2,000 samples may sound like a lot, but it's actually not powerful enough to make these types of associations. But having about 20 billion genotypes that come from these different SNPs, from all of these different 2,000 combined cases, that actually was really expensive way back in the day. Nowadays, because of the density and the speed at which we can do all of these different types of single nucleotide polymorphisms, this is on the order of about less than a cent per genotype. It is now feasible for us to do this really rapidly and cost effectively for the same amount of time and money that it used to take a long time ago. So now we're sort of at 0.1 cent, and actually we're below that now with the next generation sequencing platforms as well. So I want to just end off with the last couple of slides on a couple of different notes. Some of you have already picked papers, and some of you are starting to look at these papers for your presentations. That's a great thing. I like that, and I'm assuming that you're all going to do really well on your presentations. The biggest issue right now is how we actually do these genome-wide association studies. There has been a lot of discussion in science in the last 3 months about replication, and this has become a bioethical issue. In psychology right now, the lack of reproducibility of a tremendous number of experiments has actually led to a dramatic overhaul of how research is going to be done. If you're going to be a postdoc in the next 10 years, The way that you do research in the next 10 years is going to be different than the way that we do it right now. And those changes are coming. And a lot of this is due to replication. So we might start off with a particular study where we're looking at SNPs in a particular population, and then we need to increase or diversify the population, but we have to be able to replicate it at each of these stages. And we'll come back to that a little bit later on. When you are reviewing your papers, I'm going to ask you to follow these general criteria. And these are criteria, I'm just going to mention that when you finish this course, when you do your final exam, I'm going to ask you as one of your short answer questions, because this is a fundamental thing. What are 10 criteria that you would look for in a genome-wide association study? I'd like you to be able to, not verbatim, but give me back most of the points that I have here. And in the paper that I'm going to take you through bit by bit, I want you to see, does this paper meet all 10 of these criteria? So for example, do we really know how they measured schizophrenic individuals? Are these cases clearly defined? How do they know that these are true schizophrenic individuals? And are these the types of patients that they normally see in clinical practice? I can tell you, yes. Checklist number 1, done. It is absolutely true. Are the cases and control participants comparable to each other on important characteristics? Are they age matched? Are they sex matched? What are the criteria that they've used to match these? Those are important criteria as well. Does this particular paper that was published meet that criteria? Yes, it does. It meets it very well, and I'll take you through that paper as well. So this is actually one of the more important things. Everyone likes to think that, yes, we're really good at GWAS studies because we're so good at genome sequencing. That is absolutely true. I'm not taking anything away from the next generation or the next generation of platforms that allows us to run through genomes incredibly quickly. But the reality is, we couldn't have done this without big data and the ability to do statistics. It's really the power of statistics that has really informed us in terms of these genome-wide association studies. So when you read in the paper that the C4 SNP has been associated with an increased odd of 1.4 versus the control of developing schizophrenia, does that actually mean anything? So this is what this means. Is it of sufficient size for this odds ratio to give you an effect that actually means something in terms of the disease? In this case, yes, it does. The 1.4 is clearly an odds ratio that means that this is impactful, that if you have this particular SNP, that you are at increased risk of developing schizophrenia. So the genotyping platform, what density is that at? You have to be able to report this not only for replication, but also to see if variation is also accounted for. Are there proper quality control measures, which I'll go through in some more detail after the midterm. All of these things are things that I would like you to not only cover in your journal club presentation, but also just to know whenever you're reading these genome papers that these are things that everyone should be looking for as a critical review. Are there any replicated variants? Do they actually look at some of the previously reported results? Again, all of this gets back to statistics. Were there any corrections? You'll often read about these Bonferroni corrections. Are they appropriate? Are they suitable? Are they powerful enough? Why did they use them? All of these things have to be reported, and they have to actually make sense. And then are these also replicated in other independent population samples? This is not done often. And so this becomes an issue for some of the studies that you might be reading about. Some of you have already picked papers. Go back and see were these results actually replicated in independent population samples. Again, is it important? And if you understand what SNPs are, and if you understand haplotypes, are certain populations more likely to have certain groups of SNPs that are associated with certain features? And is that geographic origin really important? Should they be reporting all of this or not? Or did they report all of this or not? And the most crucial thing for all of us, so it's one thing to say that, yes, we have an increased risk if you have this particular SNP associated with C4, but does this actually mean that there's a functional role for what they study? And most studies fall flat there. And that's one of the big things. It could be a future direction. It could be something that you want to do next as part of your grant. But to be honest, if it's published in a high profile paper like Science or Nature or Cell, it should have sort of step number 10. There should be a functional role ascribed to whatever polymorphism they've identified. If it hasn't, that's something that you should either suggest that they do or double check that they haven't actually done it. Do you have any questions on this before we wrap up? Yes. Yeah, you could validate it. In fact, the paper that I'm going to show you validates it in an animal model. They validate it using cell culture. They validate it in a number of ways, but they have to show function. And that's the point 10 that they go through great lengths to actually go through. Anything else? Yes.
When you say show function, do you mean like describe
You don't have to describe a mechanism. So they're not 100% clear on this either. So they make a lot of intuitive leaps. So they know that the C4 SNP is involved. They know that C4, as part of the complement cascade protein, they detect it in the brain. And they know that the next step in C4 is binding to C3. And when C4 binds to the C3 protein in the complement cascade, it actually causes the reshaping of synapses. And so they don't go through all the way, but they do show that there is a functional consequence. If you knock out C4 or you modify C3, does it behave in a predictable manner if your model is correct? And so they do show that there is a functional prediction, and they're able to show that it matches what they predicted.
So just the knockout is sufficient.
In this particular case, in the way that they did it, I think it was pretty powerful. And I'll leave it to you after we go through the paper to tell me if you think that that is or not, but I do. Any other questions? Okay, so on Thursday, we're going to have the entire class devoted to a review. I'm going to give you handouts related to how you should be studying, what you should be studying, and again, highlighting what you'll likely see on the midterm. But if you have questions from any of the lectures that we've had, please bring them to class. I'm going to leave that open for you to ask me any questions as well. But if you go to the review, you should be able to get at least a 90, I'm assuming. At least a 90 on your midterm, as long as you study. You have to study. That's the biggest thing. Okay. All right. We'll see if you guys can actually meet that prediction. We'll do point number 10. We'll see if it's a functional assay that we can go back and test.